It's your boy. You already know who this is. Nah, I'm just kidding. But yeah, let's get started because I'm tired. Yeah, so I'm Vanessa and I'm talking about biochemistry. So some of the focuses of this field include molecular biochemistry, which studies enzymes and molecular processes, organisms, endocrinology, which is the study of hormones, and clinical biochemistry, which is the study of disease. So it basically studies the chemical process in living organisms, focusing on how living things obtain energy from the food they eat, the chemical basis of heredity, what fundamental changes occur in disease, and related issues. Some of the applications of this field include the biotechnology industry, pharmaceuticals, FDA, National Institutes of Health, and U.S. Department of Agriculture. And so basically, biochemistry provides knowledge on topics like genetics and heredity, and also collects data for other researchers and scientists, enabling them to make improvements to existing drugs, systems, or products. Um, Most biochemists work for government agencies, as I said before, like the FDA, National Institutes of Health, um, corporations, or university research departments. Work is generally conducted in an office setting or laboratory, and some of their duties include planning and conducting experiments, analyzing products, and developing new products, and are also responsible for presenting their findings to peers and creating grant proposals to acquire funding. So biochemistry has obvious applications in medicine, see a little hospital, dentistry, dentist, and veterinary medicine. In food science, biochemists determine the chemical composition of foods and research ways to develop abundant and inexpensive sources of nutritious products. In addition, agriculture, in agriculture, biochemists study the interaction of herbicides and insecticides with plants and pests. They examine the structure, activity, relationships of compounds, determine their ability to inhibit growth, and evaluate the toxicology effects on surrounding life. I'm very sorry. I can't talk today. So this is going as well as it's going to go. On to pay and education. So according to the Bureau of Labor, the median annual wage is about $91,000 a year, and the mean annual wage is about $105,000. Undergraduate courses generally cover topics including bacteriology and inorganic chemistry. Students may also spend significant time in the laboratory to gain a deeper understanding of biological processes. Unfortunately, a bachelor's or master's degree in biochemistry may qualify you for an entry-level position in the field. So in order to get higher jobs in the government and stuff like that, you have to have a doctorate or a PhD. So I'm going to talk about a couple of scientists who are really well known in this field. So first we have Gertie Corey, who in 1929, along with her husband Carl Corey, described what is known as the Corey cycle, which is an important part of metabolism. Basically, lactic acid forms when we use our muscles, we know that, which is then converted into glycogen in the liver. Glycogen, in turn, is converted into glucose, which is absorbed by the muscle cells. The pair continued to investigate how glycogen is broken down into glucose, and in 1938 through 1939, they were able to both identify the enzyme that initiates the decomposition and also to use the process to create glycogen in a test tube. Yeah, she ended up being the first American woman and one of, I think, 2 or 3 women in the world to receive a Nobel Peace Prize in science. So next we have Linus Pauling, who also received 2 different Nobel Peace Prizes. I think one of the first people to receive a Nobel Peace Prize in 2 different subjects, one in science and one in like advocacy, because he was like involved in protests and stuff like that. So he basically used quantum physics to understand and describe chemical bonding, that is the way atoms join together to form molecules. He worked on the structures of biologically important chemical compounds. In 1951, he published the structure of the alpha helix, which is an important basic compound of many proteins. So now I'm going to be talking about one of the most recent findings. See the date right there is June 6th. One of the most recent findings in biochemistry. By the title, you can already tell that the finding was that enzymes can adapt to cold by wiggling regions far from their active sites. And so it basically just shed light on a biophysical mechanism for low temperature adaptations. Bear with me, I can't pronounce any of these big words. Psychrophilic enzymes. Psychrophilic organisms are organisms who live in low temperature habitats. And so basically these psychrophilic enzymes tend to have more surface glycine mutations far from the active site than do similar enzymes in mesophilic organisms or enzymes. And so mesophilic organisms are organisms that live in like normal habitats like humans. Humans are mesophilic organisms. So biochemistry is really, really important in the real world because it studies how the chemical systems in organisms work, why we sweat or produce certain enzymes, and it also covers the studies on DNA, which is very important because it would help us understand why certain mutations happen in DNA, why people end up developing asthma or different illnesses like Parkinson's, Alzheimer's, stuff like that, figuring out our genetic codes. And it studies diseases and how it affects us, which helps us find cures for these diseases. So this may look very familiar to you, but this is actually a really fun and really easy experiment in biochemistry that everybody can do because it requires at-home things. Some of the things that it requires, so you need a strawberry, some isopropyl alcohol, just soap, salt, a Ziploc bag, strainer, water, measuring cups and spoons, small glass container, tweezers, a pipette, which is optional. You don't really need one because you have the measuring spoons and stuff, and a spoon. So the first step is to add 2 tablespoons of dish soap or 10 milliliters of dish soap to water. Then you stir in 1/4 of a teaspoon salt. This is going to be your extraction liquid. You place the strawberry and the extraction mixture in a Ziploc bag, as shown in step 3, and then you crush up the strawberry and mix it with the extraction fluid in step 4. Step 5, you pour it through a strainer and use a spoon to maximize the amount of mixture you get. And then in step 6, you would add 1 teaspoon or 5 milliliters of the alcohol, and you would look for the separation. In the picture, you can see that like white foamy stuff. That's the DNA. That's the strawberry DNA. And then finally, you would use tweezers to gently remove the DNA for examination. Pretty easy. I know we've done it. We've done it in biology, and it was really fun and kind of gross because it looks like mucus. So just my final thoughts. Why I chose this project and how did it deepen my understanding of science, topics related to class content. So basically, I chose this topic because it relates to something I might pursue in my future. I want to study how organisms interact with their environment and how changes in the environment affect these organisms. It was cool to see what types of things biochemists look into, such as the study of enzymes and DNA. Reading on the new discovery of enzyme development in psychrophilic organisms was insightful and honestly interesting because it shows me how incredible living organism systems are when it comes to adaptations and the organism's survival. It also went to show how certain creatures almost defy what is already presumed. The chemical reactions of the enzyme in the study remained the same despite the decrease in temperature, which we learned in chemistry affects the rate of a reaction. So that's just kind of, relating it back to what we learned about reaction rates and stuff like that, which I found very interesting. So, yeah, that's it. I'll see you around. Good talking to you.